primary obstacle targeting T-cell malignancies with the T-cell was that all of the targets that are on the T-cells are also expressed on normal T-cells. And so when you put a CAR into a normal T-cell, it just kills itself. It's called fratricide. So that's been a major issue. The second issue that has really been problematic and has really limited any development in this area is that the phenotype of the malignant T-cell in the blood is similar to a normal T-cell, so you can't separate them. So if you were to do anything to a normal T-cell, you would also be doing it, in theory, to the malignant T-cell, in theory making it resistant to therapy. So in order to get around this, we have pursued two approaches. Number one is we have used CRISPR technology to genetically edit out the target that we're going after in the malignant T-cells in the normal T-cells. And then we can put a CAR in those T-cells targeting that particular antigen. And now those cells are not susceptible, the T-cells are no longer susceptible to fratricide because they don't contain that antigen. And the other is that we've used normal T-cells from an unrelated donor or third-party donor. And in that way, we can get around the issue of contamination with malignant cells. We can also choose a young donor that hasn't been exposed to chemotherapy, so the T-cell function would be normal. Or we could even use cord blood T-cells. There's lots of advantages there. And to prevent the risk of graft versus host disease, we've also genetically edited out at the same time the T-cell receptor. So they have no T-cell receptors and they have no target, and then we put a CAR into them, and so they can't cause graft versus host disease. So we can use them. We can use cells from a single donor for many different patients, and so we can overcome many of the obstacles of CAR T-cell therapy by, number one, deleting the target, number two, deleting the TCR, and then actually we can actually use a single donor to treat many, many patients. So a single product can be used to treat as many as 300 patients instead of one product treating one patient.